Hi, and thank you for the invitation to speak today. My name is Erin Longbrake, and I'm an Assistant Professor of Neurology at Yale University. I'm going to be talking today about long-read sequencing of the 16S-23S rRNA operon to characterize gut microbial changes associated with multiple sclerosis. Here is my disclosure slide. And before we really get into the microbiome part of my talk, I want to just take a few moments to talk to you about multiple sclerosis, which may be a little less familiar. Multiple sclerosis is a chronic, often disabling autoimmune disease that attacks the central nervous system, and that's the brain, the spinal cord, and the optic nerves. And you can see in these pictures that these brighter white spots within the brain and spine represent areas of damage and demyelination caused by the disease. Normally, MS follows what we call a relapsing remitting course, where an individual who's newly acquires the disease has a sudden episode of neurologic dysfunction. And that could be blindness, it could be an inability to walk, that comes on fairly suddenly and then usually improves or completely resolves with time. The disease is relapsing, however, so after being normal for a period of time, the individual would then go on to have further relapses. And with prolonged time having the disease, usually individuals acquire disability, where they no longer recover completely, but instead they acquire neurologic disability. Again, this could be vision problems, this could be balance problems, difficulty walking. If it can be caused by the brain, it could be a symptom of multiple sclerosis. With prolonged time with the disease, individuals tend to move from a relapsing course into a secondary progressive course. So the relapsing course is characterized by waves of inflammation, whereby immune cells enter the brain and cause inflammation. The progressive course is characterized more by neurodegeneration, where nerve axons are lost and the individual, instead of having these episodes of neurologic dysfunction, instead they just slowly get worse with time in a slow but steady, relentless course. And a minority of patients follow this progressive course even from the beginning of their disease. Naturally, everyone who's ever had this disease has asked the question, what causes multiple sclerosis? And the answer that they hear from their doctors is that MS is multifactorial. We have typically a genetically susceptible individual who's exposed to some sort of environmental variable that triggers their immune system to become self-reactive. So their immune system, their immune cells actually enter the brain, the spinal cord, and the optic nerves and create damage, demyelination, and axon loss, which leads to their disability. The specific immune deficits in MS are many. There is no one cell that goes awry, and there's no one antibody that causes the disease. Instead, there are shifts in many different types of immune cells. So one type of cell that is impacted are the Th1 and Th17 cells. These are subtypes of helper T cells, and they are, very broadly speaking, pro-inflammatory. They make cytokines that perpetuate an inflammatory response, and these cells have been implicated in causing the pathology in the brain, the spinal cord, that is caused by MS. Interestingly, there are some genetic variations in the cytokine pathways used by these cells that increase the risk of MS. And self-reactive T cells, or myelin reactive, the ones that recognize and affect the nerve cells, these are more likely to have a Th17 phenotype in MS patients compared to controls, healthy individuals. Another type of immune cell that's dysregulated in multiple sclerosis are the regulatory T cells. Now, normally the regulatory T cells act in the immune system to just put the brakes on, put a damper on the whole system, and scale back any super aggressive inflammatory response. In multiple sclerosis, this subset of T cells is dysregulated. They are, instead of being more anti-inflammatory, they tend to have a more pro-inflammatory phenotype. Instead of making regulatory cytokines, they're making more inflammatory cytokines. And so overall, these regulatory T cells have an impaired suppressive function among patients who have multiple sclerosis. Another type of cell which is just emerging as being very important in MS are the B cells. And this is another type of adaptive immune cell that makes antibodies, but also serves as an antigen-presenting cell. And the B cells appear to be extremely important in MS pathology. Medications that deplete B cells actually almost totally ameliorate the relapses and the new radiologic evidence of the disease. So it's B cell depleting medications are among the most effective options for treating MS. And while there is no antibody against that causes MS, we think that the B cells may be important mainly as an antigen presenting cell that are revving up and promoting the pathogenic T cell response. That being said, the role of B cells in MS is still being elucidated, and there's much that's not understood about how these cells contribute. So I've told you that MS is caused by a genetically susceptible individual who's exposed to certain environmental triggers that lead to autoimmunity. But the real question is how? How do the genes and the environment interact? What's the mechanism by which this causes autoimmunity? And when one asks these questions, even of the foremost experts in the field, the answer that you get feels a lot like Jedi mind tricks. There seems to be a lot of hand waving that goes on. And in truth, the mechanisms are not well understood. I showed you this slide earlier that shows the natural history and the prognosis of multiple sclerosis, but not everyone follows these guidelines. So you have some people who have MS who follow a very different course. You may have individuals who have only 2 relapses over the course of their lifetime, and those could be separated by 30 years. You have patients who perhaps have 1 or 2 relapses very early in adulthood and then have no problems until they're 50 or 60, at which time they have the progressive variant of the disease. And then you have others who have an extremely aggressive disease course where they become progressive almost at the outset and continue to accumulate disability until the end of their lives. And the phases of MS are varied. You can have an MS patient using canes, walkers, and wheelchairs. You can also have MS patients who are out running marathons. And the clinical heterogeneity of the disease is really striking and not well understood. So in addition to what causes MS, we can ask the question, what causes this clinical heterogeneity of MS? What makes these patients look so different? And again, the mechanism's not understood, but one feature that stands in the midst of this equation could be the microbiome. And as a hypothesis, we might put forward that the environment changes the microbiome and shifts the immune response to lead to this autoimmune response. So let's jump into the microbiome. That's why you guys are all here, is to learn about the microbiome. So let's talk about it. We know that bacterial cells in the human body actually exceed the number of human cells, and the genetic material owned by those bacterial cells is far in excess of any of the eukaryotic DNA that we have in our bodies. We know that the intestinal commensals specifically are extremely important for digestion, for metabolizing medications, producing essential vitamins. And the gut microbiome is really important for immune system development. Without the gut microbes, the immune system never develops normally. It's a very unusual, not very effective system if there are no germs living in the intestines. Moreover, the specific gut microbes that live within us are influenced by the environment. And many of these environmental variables are similar to or identical to environmental factors known to affect multiple sclerosis. So things like body weight, vitamin D deficiency, and smoking influence both risk of MS and the composition of the microbiome. So the gut microbes do not live in a compartment. They have very important crosstalk with other organ systems, specifically the brain and the immune system. So in terms of gut-brain communications, metabolites produced by the microbiome are directly neuroactive and can act as neurotransmitters. Metabolites produced by the gut bacteria can directly stimulate the vagus nerve, among others, and provide feedback to the brain. Moreover, there's important indirect relationships between the gut microbes and the immune system, which indirectly can affect the brain, especially in situations of autoimmunity, as we're discussing today. And the immune system itself shapes which microbes end up living in the gut. The flip side of that is also true. The types of bacteria living in the gut impact how the immune system behaves. So it's a multi-directional relationship. In autoimmunity, the microbiome appears to be dysregulated, and this is seen across the board in a number of different disease states, including inflammatory bowel disease, rheumatoid arthritis, and multiple sclerosis, to name just a couple. The ways in which microbiome changes contribute to autoimmunity are still being teased out, but those mechanisms appear to include molecular mimicry. And this means that a microbe has an epitope and a 3D appearance that's recognized by the immune cells and recognized as being similar to a self-peptide. So essentially, the immune system isn't able to distinguish between the bacterial antigen and its own self-antigen. And so this is thought to be a mechanism by which autoimmunity can develop, where a microbial epitope is cross-reactive with, for example, a myelin or a nerve epitope, and the immune system develops a response to the microbe, and as a side effect, recognizes the brain and causes damage. Other ways in which the microbiome can contribute to autoimmunity, it can dysregulate the barrier function between the gut and the circulation. It can alter production of immunoregulatory molecules. So specifically, some bacteria produce short-chain fatty acids, which can include butyrate. And these are known to be immunomodulatory and promote usually an anti-inflammatory immune response. Tryptophan metabolites, among others, are also known to have direct impact on the circulating immune cells and the immune cells that are located in the gut mucosa. And so these are various ways in which the microbiome can lead to or contribute to a dysregulated immune response and an autoimmune immune response. So I'm going to go back to multiple sclerosis now. That's what we're specifically talking about today. And a number of studies so far have demonstrated that the gut microbiome is abnormal in multiple sclerosis. And there have been a handful of papers on the subject, all of which agree that the gut microbiome is abnormal in MS, but the specifics of how it's abnormal vary with the study. As you can see from this table, there's been a number of bugs implicated, and they are not the same bugs from study to study. I'm going to come back to this in just a moment as to why this is. But suffice it to say that there is agreement that the gut microbiome in MS does not look the same as the gut microbiome in a healthy individual. And this appears to be functionally important. Some studies looking at this took microbes from individuals who had multiple sclerosis or healthy individuals, and they transplanted those microbes into germ-free mice. They then gave the mice experimental autoimmune encephalomyelitis, or EAE, and that's the main animal model for multiple sclerosis, and waited to see how severe the disease was in these animals. And the mice that got EAE, who had no bugs in their intestine, that's the brown line, they actually had a milder disease course than the ones who got bugs from healthy individuals, which was more the control. So the healthy individuals are in the blue line, and that's more your quote unquote normal expected rate of EAE in this particular mouse strain. The mice who got a fecal transplant from MS patients had more severe EAE, had more severe disease, MS-like disease, than the control animals. And so that is potentially very important. Likewise, the immune cells in these animals who'd been transplanted varied. And you can see in the graph that the control animals had normal levels of regulatory T cells, whereas the animals who'd been transplanted with MS fecal microbes did not. They're in the red bar, and that indicates that their regulatory T cells were not functioning in a regulatory capacity. And if you'll recall, that is similar to what's seen in multiple sclerosis patients, which we went over a couple slides ago. This dysregulated Treg, or regulatory T cell response. So we can say that the microbiome appears to be dysregulated in MS, and that it's likely, based on animal studies, that this dysregulation can have implications for the severity of their disease. But human research is tougher than mouse research in a lot of different ways. And human microbiome research is particularly challenging in a lot of aspects. So how do we learn about the human microbiome? Well, first off, we have to recognize that humans are not identical. This is one of the beauties of animal research. You can assume that your mouse 1 is essentially equal to mouse 3 and mouse 4. Within a strain, they're genetically effectively identical. That's not the case with humans. 1, they're not genetically identical. 2, they have different environments. They take different medications. They eat different things. They were born in different ways, C-section versus vaginal birth, for example. And they have different habits. So all of these variables are known to impact the microbiome. And so any human microbiome study needs to take into account these, what we call metadata, these clinical considerations. So this is something that's very important to consider. The next point with human microbiome research is you need to decide which site you want to study. There are a lot of different places where microbes live in humans, and we have to decide which is the most important. I'm talking today about the gut microbiome, but there are also plenty of studies on the skin microbiome, the oral microbiome, respiratory or genitourinary tract. And the specific research question that you're interested in will drive which site you're looking at. After you've characterized your patients, you've decided which aspect of the microbiome you're going to study, then you have to decide your tool. And the tools for microbiome study are many and have changed over the years. So just talking a little bit about the tools for microbial taxonomy, which is the most common way that people study the microbiome. Older studies were limited to things that could be cultured, and that was very limited, as you can imagine. Not every bug that lives in the intestine is readily culturable. For sequencing technology, 16S rRNA emerged as the main way in which we understand the taxonomy of these bacteria. The 16S rRNA gene is evolutionarily conserved among bacterial species, and so sequencing just this one gene or even part of that gene can lead to taxonomic identification of your bugs. And so initially, people did full-length 16S sequencing, which took a long time, cost quite a lot of money, was very slow. This evolved into short amplicon sequencing, and we'll get into that just in a moment, and has evolved from there. So 16S rRNA sequencing of specific hypervariable regions is the standard for 16S taxonomy-based methods of characterizing the microbiome. And the 16S rRNA gene has 9 hypervariable regions, V1 through V9. And one of the things that needs to be understood about these is that not every type of short amplicon 16S rRNA sequencing is going to give you the same data. So certain of the hypervariable regions are more highly evolutionarily conserved and others mutate more frequently. The V4 through V6 regions are the most conserved evolutionarily, and the V4 region is the most similar to the average 16S rRNA gene when you sequence the whole thing. However, V4 sequencing typically cannot achieve fine detail in terms of the bacterial taxonomy. So you are limited to family, order, genus level identification. And you really can't get to species and strain level identification with V4 sequencing. If you include the V2 and V8 regions, you may be able to get a little bit more fine detail on the taxonomy, but then you run into other problems with those areas. And so I've just outlined here in this slide a couple of the benefits and fallbacks of each hypervariable region. And you see that most of them have preferences for certain types of bugs that are more easily identified in certain areas. And so the choice of region is important when considering what your tool is that you're going to study the microbiome with. So in terms of MS, the existing data are limited in a lot of different ways. The patient cohorts have been very heterogeneous. The sequencing methodologies have been variable. And we've looked primarily at taxonomy and not at bacterial function. So if I go back to this table that I showed you before and said, this looks very different from study to study, well, that could be because we're comparing apples and oranges. The methodologies used for each of these studies was very different. And so the fact that one study saw decreased Bacteroides and another saw increased Bacteroides isn't necessarily unsurprising in a way. But this does make it difficult for us to try and dial down to what's the core truth, what is the state of the microbiome in MS. And some of that we may be able to learn a little bit better based on improvements in sequencing platforms. So I talked to you a lot about the short amplicon sequencing for 16S, and most of that sequencing relies on the MeSeq or Illumina platform. This platform can sequence amplicons that are up to around 300 at a good day, probably 250 more realistically, base pairs at a time. And more third-generation sequencing technologies have changed the chemistry in such that the PacBio platform is now able to sequence 10 kilobytes at a time, so much, much longer amplicons. And the use of this platform has enabled improvements in the 16S technology so that we're no longer sequencing just the V4 region, for example. So long amplicon sequencing involving the entire 16S, the internally transcribed spacer, and the part of the 23S gene is what my group is using. And this methodology is significantly more powerful than the short amplicon sequencing in that it is able to achieve species and often strain level resolution. There are certain bugs that it won't pick up, Helicobacter and Archaea species, for example. But overall, this is a much finer granularity and allows more detailed taxonomy than any of the short amplicon sequencing. So our pipeline is to collect stool at home. We also collect detailed metadata. As I mentioned earlier, this is a very important consideration in human microbiome research. we have it shipped overnight to our lab and immediately aliquot. And then when we're ready to process, we do so in batches. Isolate the DNA, achieve the sequences, demultiplex the sequences. We use data2 to denoise the data and then align against an Athena database that's a modified version that includes the internally transcribed spacer and the 23S gene as well. Just a short look at our demographics. I'm going to summarize this to say that we have a little over 30 MS patients, and we have longitudinal samples on most of these patients, both at baseline and then after they start immunomodulatory therapy. And we have healthy controls to match them. And we've collected detailed information about these patients, their diets, their stool habits, their travel history, just everything we could think of that might potentially impact the microbiome. We, again, as I showed you our pipeline, just looking at the mock community reads, we're just demonstrating quality of this pipeline. You can see that before the data2 denoising step, we do have a lot of read counts, which gets decreased as expected when the errors are filtered out with the data2 step. And the important things to note is that the post-data2 sequences that have more than 10 aggregate reads over the course of the entire data set is almost identical to that that's expected based on manufacturer-provided specs about the mock communities. And that after data2, we have essentially 100% of our reads are assigned to the expected species. So we feel very confident this is a high-quality way of analyzing these data. We then are going to come back to our specimens, and I'm just going to take just a quick moment to talk about ways of measuring the microbiome. And we look often at alpha and beta diversity. So alpha diversity refers to diversity within a sample, how many species can be detected within a given specimen. You can also talk about beta diversity, which is diversity between samples. So the top set of circles has high beta diversity because many species are different between these two organisms or these two sets of data. Whereas the lower circle would refer to a low beta diversity, even though there's a lot of different bugs in that lower right circle. They're almost all shared with the circle on the left. So the beta diversity there would be lower. And in our data set, the alpha and beta diversity of MS patients are significantly different compared to the healthy controls. Overall, the alpha diversity was decreased amongst our MS patients. And this was most pronounced before they started immunomodulatory therapy. The beta diversity, if anything, was increased in the in the MS patients at baseline. And we then wanted to look at the overall underlying populations making up this change in diversity, because the alpha and the beta diversity tell us nothing about the specific bugs, only that there's a difference. And we did an analysis of the composition of microbiomes, which is a data technique that's designed for microbiome research, just looking at overall which sets of bugs are different. And we identified Bacteroides as relatively enriched in patients who had multiple sclerosis. So we then wanted to look at which specific bugs were enriched, and we also wanted to examine the impact of immunotherapy on the gut microbiome. So patients with MS typically are treated with immunotherapy. And in our cohort, these are actually, all of these patients were treated with B-cell depleting immunotherapies. And many medications, including some other immune-modifying medications, do impact the composition of the gut microbiome. And we were curious whether that was part of how these mechanisms were, how these medications were acting in MS. And so we looked at the species level identification of these Bacteroides. Again, this was identified at the genus level as being enriched in MS. Here you can see some of the specific species which appeared to be enriched in the MS patients compared to the healthy individuals. And interestingly, you can see that the most of these species appear to decrease, or the trends is more for a distribution that more resembles the healthy controls as the individuals are treated with immunomodulatory B-cell depleting medications for longer periods of time, which is really interesting, and we want to flesh this out further. So future directions for us is that we are still increasing our cohort size and the extent of our longitudinal follow-up with these patients. I told you that we'd collected a lot of clinical metadata into this, and that this collection of clinical metadata is extremely important for human microbiome research. And so we are still incorporating these clinical metadata into our analysis. We also are planning to expand this data set into multi-omic research and look also at the metabolites produced by these microbes. And that will be yet another very important aspect of how the microbiome is acting. So it likely won't be enough just to know which bugs are there and which are not. We'll also need to know what they're doing. What are the metabolic implications or the functional implications of this change and this dysbiosis that we see in autoimmune disease. So that is my talk for today. I'd like to give special acknowledgement to Bill Ruff, upper right picture, to Gunit Janda and to Ina Cohen, my students, who have just done most of this work, and it's just a phenomenal job from all three of them. Really made this work possible. Also acknowledge David Hafler, my mentor, and his lab, and my funding sources. And with that, I would like to thank you for your attention. Any questions can be directed towards me via the platform. Thank you so much.